Hello, I'm Yuri Lee. I'm from North London College School in Jeju from South Korea. Today in this presentation, I'm going to introduce my invention titled A Novel Method for Enhancing Crop Yield with Plant Growth Promoting Bacteria. So first of all, what is sustainable agriculture? Sustainable agriculture is a farming in sustainable ways, which means meeting society's present food needs without compromising the ability of future generations to meet their needs. Plant growth promoting bacteria may be one of the keys to lead to sustainable agriculture practices. Two different types of bacteria are chosen to be inoculated in Arabidopsis. The purpose of PGPB stimulates plant growth involves the availability of nutrients originating from genetic processes. P. florensis is used as root protection, phosphate solubilization, and the production of cytohormones. B. japonicum is used for biological nitrogen fixation. It brings the nitrogen in the air and converts into nitrates for the plants. These candidates are used because they colonize roots and create a favorable environment for development and function. For research method, 4 steps were conducted in this research. First, plant growth conditions were optimized. Secondly, PGPB inoculants were prepared. Thirdly, PGPB were applied to plants. Lastly, the plant weight was measured and the data analysis was performed. Next, I will explain the result of this project. The top right image shows the Arabidopsis plant on their control environment. The image on the left is taken after 2 weeks and the image on the right is taken after 3 weeks. On the left middle section, the final result is shown. I prepared 4 different inoculation samples, MOG, which is a sample without bacteria inoculation. PF indicates the sample inoculated with only P. florensis. BJ indicates the sample inoculation with only B. japonicum. PF plus BJ indicates the sample inoculated with both bacteria. The inoculation concentration was prepared as 1 and 100 concentration, where 1 is 10 to the power of 9 CFU per milliliter. Lastly, 2 conditions were prepared: normal water conditions, which is water every 2 days, and just stress conditions, which is water every 7 days. After the samples were grown for 5 weeks, the weight was measured for each condition. The average of plant weight was analyzed in this graph. Interestingly, the left graph result shows that the plant weight was increased only when BJ 100 was inoculated in normal water condition. This result indicates that when water is sufficiently provided, only B. japonicum inoculated samples increase the growth. Since B. japonicum is known to be nitrogen-fixing bacteria, the growth of Arabidopsis may be enhanced by extra nitrogen supplied by the bacteria. However, the right graph shows an interesting result. When the Arabidopsis was grown in drought stress conditions, inoculating with B. japonicum alone was not sufficient to enhance the growth of the Arabidopsis. Instead, P. florensis alone and combination of both bacteria significantly increased the growth of Arabidopsis as shown in the right graph. This result was consistent with the previous study that P. solubilizing bacteria supports water supply for plants and promotes the plant growth. Not only the biomass changes, but also stress tolerance was increased by PGPB treatment in my experiment. The result will give an idea of how PGPB can function to stimulate plant growth and can be applied to increase the crop production yield in sustainable ways. Thank you.